The human race is expanding rapidly. More and more humans are born every day. Where will mankind be in 1,000 years? How will today's knowledge have subsisted? What will have remained of us? The answer is simple. Data, bits and bytes, zeros and ones, will surround us every day and define life on Earth even 1,000 years from now. The human race produces tremendous amounts of data every day, which soon could be too much for our storage supply. By 2025, 175 zettabytes of data are predicted to be produced, whilst only 20% of it can be stored. Hard drives and magnetic tapes will soon no longer provide the data capacity and durability to store crucial information. But what if all of our information gets lost? Loss of data from the health or finance sector could pose a serious risk to infrastructure and safety. Loss of cultural and historical data will leave a void in human history. How can we protect the future by preserving the present? The solution is to be found inside ourselves, in every cell of our body, DNA. It is one of the most stable, sustainable, and dense storage methods. DNA is a molecule built of a sequence of 4 bases. In nature, genetic information has been stored for millions of years, making it the oldest code of life. However, not only genetic information can be saved in DNA. Thanks to developments in synthetic biology, any information can be archived in artificial DNA. Hundreds of petabytes can be stored in a single gram of DNA. With that, you could fit the information of the whole internet into an ordinary bucket. DNA is a natural molecule which, in contrast to hard drives and magnetic tapes, does not require limited resources such as semiconductors. Encapsulated in silicon dioxide, DNA can be archived for millennia among a wide range of conditions. In a couple of years, you might be able to store your most valuable information on a DNA data stick. And who knows whether mankind will still be living on Earth? For the colonization of other planets, we will need to transport our data as efficiently as possible. With our skills in synthetic biology, we will boost a new revolution in DNA storage systems.
With the need for improved methods to save vast amounts of data for long term, we aim to develop a system for enzyme-based DNA synthesis. This includes immobilization of DNA, followed by the synthesis of a preset sequence. The sequence to be generated gets dictated by an algorithm, converting digital information into DNA and vice versa. The terminal Deoxynucleotidyl transferase, short TDT, is an enzyme that catalyzes the addition of nucleotides to the 3' terminal end of DNA. It is one of the few DNA polymerases that does not need a double-strand template for synthesis. Instead, TDT can extend single-stranded DNA by randomly adding nucleotides to the strand. Another unique feature of the TDT is its ability to use a wide range of cofactors, which alter towards incorporating certain nucleotides. So, our vision for this project was to connect single-stranded DNA immobilization with custom-built, affordable hardware, all supported by the improved software with proof-viewing capabilities, to generate a unique approach to DNA synthesis.
The entirety of the project is a lot to grasp. To get a better understanding of our project's most essential parts and methods, let me walk you through an overview. The foundation of our project is the wet lab. Instead of a specific synthesis, in which each base would store information, we used a semi-specific approach, where information is only stored in the transitions. This means that the TDT may add a random number of nucleotides. Our experiments were adapted to this approach, and the generated data was used to feed into models. These could in return give us valuable information for future experiments. The software could then be tailored to our needs, translating digital information into a DNA sequence and vice versa. For now, let us focus on our approach in the lab. As our key component is the enzyme TdT, its characterization was our main concern. To do so, we chose agarose gel electrophoresis for easy analysis. However, as we could only make estimations, we soon expanded our repertoire and included capillary electrophoresis. This allowed us to obtain more accurate size determinations and information about length distributions. The next logical step was sequencing. After performing a PCR, the amplified double-stranded samples could then be analyzed via nanopore sequencing or examined with Sanger sequencing. As we gained a deeper understanding of the TdT, we worked on different approaches to immobilize our DNA primers. This was needed for another major part of the project, the hardware. So now that we understand the fundamental structure of our project, let's talk about the laboratory, modeling, and software results we gathered throughout the months.
As we have seen, the first step was to characterize the TdT for its conditions. Thus, we established an agarose gel electrophoresis protocol for reliable length estimation of our elongated strands. Please note that we analyzed single strands unless amplified by a PCR, but used a double-stranded DNA marker. Therefore, the resulting bands are always relative size estimations. In a standard TdT reaction with cobalt as a cofactor of choice, we found that the nucleotide incorporation efficiency is highest for T, followed by C, A, and G. Experimenting at 37 degrees revealed the same incorporation efficiency. Additionally, we could observe sharper bands, suggesting an improved size distribution, especially for C and G. Nevertheless, we choose to work at room temperature, as it allowed us to design simpler hardware, make it more accessible to be rebuilt by fellow scientists. Adjusting the elongation speed was a crucial aspect for our system. That's why we investigated various parameters and found that adjusting the cofactor concentration in the reaction buffer is a potent way to do so. We decreased the cofactor in defined steps, which resulted in steadily reduced elongation. Next, we varied incubation time, since it's the most straightforward way of dictating the obtained strength length. The first successful test had a large timescale, ranging from 1 to 90 minutes. The obtained results revealed rapid nucleotide incorporation within the first minute already. However, gel electrophoresis has a limited resolution, so we switched to capillary electrophoresis and analyzed the incorporation of each nucleotide over 2 minutes. Generally, we could observe that the size distribution increases along with the reaction time. Again, elongation with T results in the longer strands, whereas in this case, C, A, and G were all elongated between 10 and 20 nucleotides.
Our next achievement was the immobilization of biotin-labeled primers onto streptavidin-coated magnetic beads. As multiple experiments revealed that the reaction works best when the primer-bead conjugate is in solution, we designed a non-permanent immobilization approach. This involves a plastic cover that creates a barrier between the magnet and the magnetic beads. That way, the magnet can be used to transfer the beads across the tubes, but can also be removed during the reactions, enabling elongation to take place in solution. We call this the dip method. To verify immobilization, we set up an experiment in which every step of this dip method can be analyzed. On an agarose gel, we observed that most primer was conjugated and immobilized, so the washing steps do not disrupt the conjugation or immobilization, and the elution appears to contain all of the primer. As successful immobilization was verified, we focused on generating base transitions in cyclic elongation approaches. In our first trials, we could already retrieve 80% of encoded information from short sequences. This was achieved by a manual cyclic synthesis, followed by PCR, nanopore sequencing, and subsequent evaluation with our software.
How the hardware will operate, for example, how long a circle will be, was explored by our model. It helped us a lot because we could test different conditions before actually implementing them in the lab, which saved a lot of time. The model consists of a reaction network, which is modeled by a system of coupled differential equations. It allowed us to get some general insight into the kinetics of the reaction system, but also tackle the optimization problems we are facing. By doing parameter analysis, we could find ideal reaction conditions, like the concentration of nucleotides and enzymes needed to achieve a special reaction time. We also found that nucleotide concentration only has a small influence when the reaction time gets longer. To further improve our model, we used wet lab data from capillary electrophoresis and did the nonlinear regression using MATLAB. The data obtained from the regression allowed us to create an even more accurate model.
To close the circle from digital data to DNA and back to data, we developed a software tool called DNA Utils that can encode information in DNA and recover it from sequencing data. Encoding involves two steps. First, the software needs to convert the digital binary data into ternary data. Then it translates this ternary data into a DNA sequence. As mentioned before, our approach is to encode the information into the changes from one nucleotide to another. This is also the reason why we use a ternary code. From each nucleotide, 3 others are reachable. Decoding works just as encoding but backward. However, there's one additional step involved. We first need to retrieve the original DNA sequence from all the sequencing data. To do so, our software first removes the primer and other strands added for synthesis and sequencing. Secondly, the homonucleotides created while synthesis must be translated back to single nucleotides. This step also includes a type of error correction. Since nanopore sequencing creates thousands of DNA strands, our software can compare all the different sequences. This makes it possible to robustly recreate the original sequence, which then can be decoded back to digital data.
To make DNA the future of data storage, it is necessary that large quantities can be produced quickly. For this purpose, we have developed a machine that automates the otherwise time-consuming process in the laboratory, DIPSY. It consists of a revolver in which the reaction tubes are placed and a vertically movable clamp. On it, a rod was attached to immobilize the primers. A sequence can be entered via a web interface, which the machine then synthesizes through the DIP method.
To drive innovation further, it is crucial to exchange ideas with like-minded people. It was a great opportunity to meet renowned scientists like George Church, Stephen Benner and Sebastian Palluk in order to discuss our project and goals. All of them were fully committed to sharing ideas and resolutions with us, which boosted our creativity in the course of our project. George Church helped us improve our hardware and motivated us to take risks to achieve the unachievable. As a pioneer in enzymatic DNA synthesis, Sebastian Palluk gave us valuable tips for working with the TDT and was always a source of inspiration in the development of our project. And last but not least, Stephen Benner encouraged us to use unnatural bases to both increase data density and prevent the formation of secondary structures. We were lucky to meet many more enthusiastic scientists to help us on our journey.
Not only were we mentored about science by specialists, but we also slipped into this role during our human practice projects. Communication of science is a crucial part of the iGEM journey. So we took part in an Erasmus Plus program organized by the iGEM team Trace, where we examined the question of how to communicate science to the public. That's why we prepared different stage plays about climate change, vaccination and synthetic biology, and presented them to the public as non-typical forms of science communication. We also shared our enthusiasm for science by giving students some experience of lab work. This way, we wanted to inspire and bring them in touch with the field of biotechnology. Thus, our team extracted DNA from strawberries and created lava lamps with the youngsters. But we didn't stop there. As part of a collaboration with numerous iGEM teams, we designed a postcard that aims to communicate science via visual art. To spread our project even more, we published an article in the magazine Biospectrum. We also reached out to the City Archive in Aachen as a potential user of our DNA data storage. Through this visit, we were able to learn about the problems with the current long-term storage and try to address them in our work.
Throughout the whole competition, we have put extensive efforts into ensuring high safety standards, both in terms of COVID-19 and safety at work. To achieve all this, all of our team members carefully followed the recommended hygiene concepts, plus we limited ourselves to working only in pairs of 2 or even alone. Additionally, we put a lot of work into carefully writing down standard operation procedures, short SOPs, containing information on the methods, devices, chemicals, risk areas and proper disposal. This did not only open up a safe and free workflow, which every team member could follow, it also allowed each one of us to jump right into the lab and safely perform methods even for their first time, thus saving hours of work and communication.
For our journey, we had the vision of improving enzymatic DNA synthesis to a point where it is more efficient. Because of the limitation of time and resources, we could not put each of our ideas into effective practice. However, the limitation of this competition shall not hold us back from expressing our concepts. Once the fundamental framework for DNA data storage is established, we hope that our research can push DNA synthesis further, make it more effective and practically applicable. With Storagene, we achieved extraordinary success. We were able to prove that semi-specific enzymatic DNA synthesis is also possible with non-permanently immobilized DNA strands. Additionally, we have created a unique DNA encoding software that is straightforward and open source, so everyone can work on it.
As we believe that biotechnology deserves broader general attention, we organized and took part in several scientific communication activities to spread the word about both iGEM and biotechnology as a field of study among students. Reaching out to organizations, companies, as well as experts did not only give us meaningful insight regarding project realization, it has also helped us to further develop our soft skills. By combining our knowledge, we created a hardware that is made of 3D printed and standard building parts, allowing easy replication. We laid the foundation for the beginning of a new era. How will our life look like if DNA data storage succeeds? We cannot wait to find out.
We are iGEM Aachen, and we are the future of DNA data storage.